Hello friends, welcome in this hematological section of human physiology. I am Dr. B.K. Prasad. Today I will talk about this WBC. What is WBC? You know that WBC is also known as white blood cell, or white blood corpuscle or leukocyte. Total leukocyte count is 4 to 11,000 per cubic millimeter of blood. This is 4 to 11,000 per cubic millimeter of blood. When it decreases, number decreases below 4,000, then it is known as leukopenia. Or when it increases above 11,000, then it is known as leukocytosis. White blood cell 4,000 to 11,000 per cubic millimeter of blood. Now WBC is, when stained with Leishman stain, WBC is classified into two groups. WBC is classified into two groups. Granulocyte means granules are present in their cytoplasm, and second is agranulocyte. Agranulocyte means granules are not present in the cytoplasm of the WBC. Granulocytes are divided into neutrophils. Neutrophils, Eosinophils, and Basophils. Agranulocytes are Monocytes and Lymphocytes. Is clear? WBC is divided into two parts, granulocyte and agranulocyte. Granulocyte has granules in their cytoplasm. Agranulocytes do not have granules in their cytoplasm. Special granules. Here special granules are present. Here special granules are not present. That is termed special granules. Now we will learn about this one by one. Coming to neutrophil. Neutrophil size is 10 to 14 micron. Cytoplasm contains granules, fine granules. Cytoplasm contains fine granules, red and white both. Red and white granules both are present, and nucleus is multilobe. 2 to 4 are present, and here in the cytoplasm there is fine granules are present. Both types of granules are present. Eosinophilic granules, that is red granules, as well as blue granules, that is basophilic granules. Therefore it is known as neutrophil, and its another name is polymorphonuclear cells. Polymorphonuclear cells means these are parts of nucleus, and this is polymorphonuclear cells or polymorphs. It is also known as polymorphs. Cytoplasm contains the nucleus, character of nucleus. This is a neutrophil. This is neutrophil. Now, eosinophil. Size is 10 to 14 micron. cytoplasm contains granules. Cytoplasm contains granules which is red in color, that is eosinophilic granules, and these are coarse granules. And nucleus is bilobe, like spectacles. This is just like this. Only eosinophilic granules, red granules are present here. This is eosinophil. Cytoplasm contains granules, nucleus is bilobe, and percentage is 2 to 6%. In neutrophil it was 50 to 70%. 70, what is the meaning? That when you will count 100 WBC, then you will get 50 to 70% neutrophils. When you will count this 100 WBC, then you will get 2 to 6% of this eosinophil. Now basophil. It is clear? Now basophil. Basophil has basophilic nucleus is bilobe, but it is not very clear, just like this. Size is 10 to Basophils. Size is 10 to 14 micron. Cytoplasm contains many coarse bluish granules, that is basophilic granules are there. And these granules are covered. These granules cover the or mask the nucleus. This is basophil. Size is 10 to 14 micron, and cytoplasm contains blue granules. Nucleus is bilobe, and its percentage is 0 to 2%. When it is more than 2%, then it is called basophilia. Now monocyte. Monocyte is, say monocyte. Monocyte is the largest cell of WBC. What to say about WBC? It is largest cell of the blood. This is 10 to 18 micron, 14 to 18 micron. Nucleus is kidney shaped. Or being shaped, and there is no special granules in the cytoplasm. This is nucleus is this kidney shaped nucleus is there. Kidney shaped nucleus is present here. This is monocyte. 2 to 10%. Okay, nucleus is kidney shaped and cytoplasm is clear, no granules are present, and 2 to 10%. When it decreases below 2%, it is monocytopenia. When more than 10%, it is monocytosis. Okay, now lymphocytes. Lymphocytes are of two types. First is, say, a small lymphocyte. Small lymphocytes have large nucleus. Only rim of the cytoplasm is there. It is T lymphocyte or a small lymphocyte. Another is, say, large lymphocyte. Its size is 7 to 10 micron. Size is T lymphocyte. Size is 7 to 10 micron. A large lymphocyte or B lymphocyte size is more, that is 10 to 14 micron. 10 to 14 micron. Nucleus is a small. Here, nucleus is a small. Nucleus is a small in large lymphocyte, and it is 20 to 30%. Lymphocytes are 20 to 30%. What are the characteristics of lymphocyte? You will recognize lymphocyte by round nucleus, clear cytoplasm. A small lymphocyte is of 7 to 10 micron, large lymphocyte is 10 to 14 micron, and it is 20 to 30%. Lymphocytosis, what is lymphocytosis? Lymphocytosis is when lymphocyte count is more than 30%, and lymphopenia when it is below 20%. When it is below 20%, it is known as lymphopenia. These are the cells of WBC. In up to now, just sum up. What is leukocytosis? Leukocytosis means when leukocyte count is more than 11,000 per cubic millimeter per milliliter. Leukopenia when it is less than 4,000 per cubic milliliter. And then neutrophilia, neutropenia, it is clear? Neutropenia, eosinophilia, basophilia, monocytosis, monocytopenia, here on this side, lymphocytosis or lymphopenia. Neutrophilia means neutrophil count is more than 70%, neutropenia less than 40%, eosinophilia more than 6%, eosinopenia less than 2%, basophilia more than 2%. That is, there is no basopenia because its count is 0 to 2%. Basophilia, monocytosis more than 10%, monocytopenia less than 2%, lymphocytosis more than 30%, and lymphopenia more than 30% and less than 20%. This is about WBC character. Now formation of formation of leukocytes. Leukocyte formation is known as Lecopoesis. Formation of leukocyte is known as Lecopoesis. Formation of WBC or leukocyte is Lecopoesis. Lecopoesis, site of leukopoiesis is red bone marrow. In red bone marrow, there is a stem cell. In a stem cell or pluripotent cells, or hemocytoplasm. This myeloid stem cell, then lymphoid stem cell. From myeloid stem cell, RBC formation occurs, WBC and platelets. In WBC, granulocyte and monocyte. Lymphocytes. Now it depends upon hormone, that is erythropoietin, then thrombopoietin. This will cause development of RBC forming cells by erythropoietin, thrombopoietin platelet forming cells, and WBC granulocyte series. In granulocyte series, myeloid stem cell, then monoblast, interleukin 3, interleukin 5, and agranulocyte colony stimulating hormone, agranulocyte colony stimulating factor. This will cause formation of monoblast to pro monocyte, then monocyte, and this monocyte will come into bloodstream, from where it will migrate to the tissues, and in tissues it forms macrophage. In macrophage, when it goes into the CNS, it becomes microglia. When it goes into the liver, it forms Kupffer cell. In lung, it is alveolar macrophage, and in bone, it becomes osteoclast. It is function of macrophage. What is the function of macrophage? If a macrophage is very phagocytic and antigen presenting cell. This is about macrophage. Now, from this monocyte is formed. Second is myeloblast. Granulocyte colony stimulating factor IL-3 and IL-5. Second is monoblast series and myeloblast series. Myeloblast cell is stimulated by granulocyte colony stimulating factor GCSF. Granulocyte colony stimulating factor IL-3, interleukin 3 and interleukin 5. In this, now it will develop into three things. Say neutrophilic, promyelocyte, then neutrophilic myelocyte, then neutrophilic band cell, and finally neutrophils. What is neutrophil band cell? Single. Nucleus is not divided into lobes. Single. Then it is band cell. So neutrophils, myeloblast develops into neutrophilic promyelocyte, then neutrophilic band cell, and finally neutrophil. From myeloblast, one thing, another basophilic promyelocyte, and it will develop into basophilic myelocyte, and then basophilic band cell, and finally basophils. Band cells, in band cell there is nucleus is not lobe, it is just U-shaped. Nucleus is U-shaped. It is band cell, not lobe. And during development, it will develop into, it fold upon, and this will form the bilobe nucleus. Then eosinophilic promyelocyte, eosinophilic myelocyte, eosinophilic band cell, and finally eosinophil. This is the development of WBC. Now, from the myeloid cells, myeloid stem cell, due to stimulation by thrombopoietin. By stimulation of thrombopoietin, thrombopoietin is secreted mainly from kidney and to some extent by liver. From thrombopoietin like erythropoietin, then it will form megakaryoblast, megakaryocyte is large cell and it fragments, cytoplasmic fragments, its cytoplasmic megakaryocyte here it is megakaryoblast or megakaryoblast 2 megakaryocyte then megakaryocyte, it is a very large cell and this will fragment and its fragment, cytoplasmic fragments will form platelets. Platelets are without nucleus. This is about development of this WBC and platelet. Now, in short, functions of these cells. In short, this neutrophil functions of neutrophils. Neutrophils function is phagocytic. Eosinophils anti-worm functions. Basophils contains heparin and histamine. Heparin is natural anticoagulant and histamine is involved in inflammation. Neutrophil phagocytic action है and it kills the invading organism by oxidative process and aerobic process and non-aerobic process. Okay, now eosinophil monocyte, we talked about monocyte. In the bloodstream it is circulating and migrate into the tissues where it will remain in as macrophage, or in CNS it is known as macroglia, in lung it is alveolar macrophage, liver Cooper cell, and in bone osteoclast. Function is phagocytic. Its function is also phagocytic and antigen presenting cell. Now lymphocytes. Lymphocytes are involved in chronic infection. This is also involved in killing the bacteria. T cell, it is two types of cells are there, B cells, B lymphocyte. B lymphocyte, due to stimulation by antigen, it is transformed into plasma cells. Plasma cells will form different types of immunoglobulin, IgM, IgG, IgE, IgA, and IgD. This is transformed into plasma cells and this form 5 types of immunoglobulin, synthesize 5 types of immunoglobulin, and this is involved in immunity. This is involved in humoral immunity. And then T lymphocytes, thymus dependent. And this T lymphocytes, after production, after formation, it goes into the thymus, and there it is the T lymphocytes matures, and there are different types of T lymphocytes. First, helper T cells help B cells, then cytotoxic T cells. Cytotoxic T cells kills the bacteria uh, due to apoptosis. And other types are memory T cells. These cells are modified into T cells, T lymphocytes modified into different types of T cells, helper T cells, cytotoxic T cells, memory T cells, suppressor T cells, etc. This is all about the WBC. I think it is helpful for you. It is if you think that it is of some value, then please like the video and share it with your friends and colleagues. Thank you very much.